Dear students, we are going to discuss about cell division, comparison between mitosis and meiosis, crossing over. First come to the introduction. Cell division is a complex phenomenon by which the meso cell divides and forms new daughter cells. It is the final and microscopically visible phase of an underlying change that has occurred at molecular and biochemical levels. All cells are produced by the division of the pre-existing cells. Continuity of life depends on cell division. It becomes a means of reproduction in unicellular organisms. Thus, it is central to life of the cell and is essential for the perpetuation of the individual. There are two types of cell division in eukaryotic cells. They are number 1, mitosis, and number 2, meiosis. Now, let's first discuss about the mitosis. Mitosis is the type of cell division in which a parent cell divides into two daughter cells with the same number and type of chromosomes as are present in the parent cell. It occurs in somatic cells and it is meant for the multiplication of cell during embryogenesis and blastogenesis of plants and animals. It is important for the replacement of cells lost to wear and tear and for wound healing. The process of mitosis involves a series of events both in the nucleus and in cytoplasm. Thus, it can be divided into two stages: A. Karyokinesis or nuclear division, and B. Cytokinesis or cytoplasmic division. First, karyokinesis or division of nucleus. The process of karyokinesis includes the division of nucleus into two daughter nuclei. The nucleus undergoes a number of complex but well-organized and regular steps. The process of karyokinesis can be separated into 5 phases. They are Roman 1, prophase; Roman 2, prometaphase; Roman 3, metaphase; Roman 4, anaphase; and Roman 5, telophase. Now, first discuss about the prophase. The appearance of thin thread-like condensing chromosomes marks the beginning of prophase. The sister chromatids of each chromosomes are held together by a centromere. As prophase progresses, the chromatids become shorter and thicker. The chromosomes approach the nuclear envelope, causing the central space of the nucleus to become empty, and nucleolus gradually disintegrates. In the cytoplasm of the early prophase, the centrioles become duplicated, and each centriole starts migration towards the opposite poles. Between the separating centrioles, spindle fibers are formed. In cells of hair plants, however, spindle fiber forms without the aid of centrioles and lacks esters. The disappearance of nuclear envelope marks the end of prophase. Now let's come to the prometaphase. The breakdown of nuclear envelope signals the commencement of prometaphase. The spindle fibers capture the kinetochores of the chromosomes and try to align them on the metaphase plate. As the metaphase approaches, balanced bipolar forces hold chromosomes on the metaphase plate. Now, let's come to the metaphase. During metaphase, the sister chromatids are still held together by the centromere and occupy the equatorial plate. The kinetochores of the two sister chromatids face opposite poles. The metaphase stage continues till all the chromosomes are arranged at the metaphase plate. Now, let's come to the anaphase. The anaphase begins abruptly with the synchronous splitting of its chromosome into its sister chromatids and becomes chromosomes. The daughter chromosomes move apart and migrate towards the opposite poles. During their migration, the centromeres remain foremost and the arms of the daughter chromosomes directed towards the equator, so that the chromosomes characteristically appear U, V, or Z-shaped. Now, let's come to the last stage, the telophase. The telophase begins with the migrating daughter chromosomes reach the opposite poles. During it, the nuclear envelope reassembles around its group of chromosomes to form two daughter nuclei. The mitotic apparatus, except the centrioles, disappear. High viscosity of the cytoplasm decreases. The chromosomes resume their long, slender, extended form, and RNA synthesis restarts, causing the nucleolus to reappear. Now let's come to the cytokinesis. The cytokinesis begins with the unfolding of the chromosome and formation of nuclear envelope. The cytoplasm constricts in the equatorial region, and this constriction is accentuated and deepened until the cell divides. During it, the cytoplasmic components, including the mitochondria and the Golgi complex, are distributed to the daughter cells. Now, let's discuss about the meiosis. It is a type of cell division in which the diploid parent cell duplicates its chromosomes only once but divides twice in succession, resulting in the production of 4 cells containing half the number of chromosomes present in the parent cell. It occurs in germ cells of sexually reproducing organisms. The 2 successive divisions take place without an intervening interface. In the first division, a diploid parent cell produces 2 haploid cells and is known as reduction division. The exchange of genetic materials between the 2 non-sister chromatids occurs in this division. In the second meiotic division, the 2 haploid cells divide mitotically and result into 4 haploid cells. No pairing of homologous chromosomes and exchange of genetic material takes place in the second meiotic division. Both the meiotic division 1 and the meiotic division 2 are further divided into different stages. First, discuss about the meiotic division 1. Meiotic division 1 is divided into 5 stages. Number 1, prophase 1. Number 2, prometaphase 1. Number 3, metaphase 1. Number 4, anaphase 1. And number 5, telophase 1. Prophase 1 is the longest stage of the meiotic division and includes the following sub-stages. Roman number 1, proleptotin. In the proleptotin, the chromosomes are extremely thin, long, uncoiled, and closely resembles with the early mitotic prophase. Roman number 2 is the leptotin. In the leptotin, the nucleus has increased in size and chromosomes have become more apparent. Frequently, the chromosomes have a definite polarization and form loops whose ends are attached to the nuclear envelope at points near the centrioles. Roman number 3, zygotin. The first essential phenomenon of meiosis, that is synapsis, occurs in the zygotin. The homologous chromosomes become aligned and undergo pairing. The pairing may start from within the chromosomes and continue towards the end of the chromosome. In other cases, fusion occurs simultaneously at various places along the length of the chromosomes. The pairing is associated with the formation of highly organized but complex protein called synaptonymal complex. Now let's come to the pegchitin. In pegchitin, the chromosomes contract longitudinally and they become thicker and shorter. In the middle of the pegchitin, its homologous chromosome splits lengthwise to form two chromatids. In the late pegchitin, a line of separation perpendicular to the plane of pairing appears and four chromatids become visible. The exchange of genetic materials between the chromatids of the homologous chromosomes, known as crossing over, takes place during this substage. The fifth one is diplotin. The separation of the paired homologous chromosome, known as desynapsis, starts at diplotin. The synaptonymal complex appears dissolved, leaving participating chromatids of the paired homologous chromosomes physically joined at one or more discrete points called chiasmata. The last substage is the dikinesis. In the dikinesis, the bivalent chromosomes become more condensed and evenly distributed in the nucleus. The nucleolus detaches from the nucleolar organizer and ultimately disappears. Nuclear envelope breaks down, the chiasmata move from the centromere towards the end of the chromosomes, and the intermediate chiasmata diminish. The chromatids remain connected by the terminal chiasma, and this exists up to the metaphase. Next stage is the prometaphase. In the prometaphase, the nuclear envelope disintegrates and spindle fiber arises in between the two centrioles which occupy the opposite poles. The chromosomes become greatly coiled, attached by the spindle fibers at their centromere and hold them at the equatorial plate. Now let's come to the third stage, that is metaphase 1. During metaphase 1, the spindle fibers are attached to the centromere of the homologous chromosomes. The centromere of each chromosome is directed towards the opposite poles. The repulsive forces between the homologous chromosomes increase greatly, and the chromosomes become ready to separate. Now, let's come to the anaphase 1. At anaphase 1, homologous chromosomes are freed from each other, and due to the shortening of the chromosomal fibers, each homologous chromosomes with its two chromatids and undivided centromere moves towards the opposite poles of the cell. The actual reduction in the number of chromosomes occurs at this stage. The last stage of meiosis 1 is the telophase 1. The arrival of a haploid set of chromosomes at each pole defines the onset of telophase 1, during which nuclei are reassembled, the endoplasmic reticulum forms the nuclear envelope around the chromosomes, and the chromosomes become uncoiled. The nucleolus reappears, and thus two daughter nuclei are formed. After the karyokinesis, cytokinesis occurs, and two haploid cells are formed. Both the cells pass through a short resting interphase. During interphase, no DNA replication occurs. Now, let's come to the meiotic division 2. The second meiotic division is actually the mitotic division, which divides its haploid meiotic cell into two haploid cells. The second meiotic division includes following four stages. Number 1, prophase 2. In the second prophase, each centriole divides into two, and they migrate to the opposite poles. The spindle fibers are arranged at the right angle of the spindle of first meiosis. Nuclear membrane and the nucleolus disappear. The chromosomes with two chromatids become short and thick. Next stage is the metaphase 2. During metaphase 2, the chromosomes get arranged on the equator of the spindle. The centromere divides into two, and thus each chromosome produces two daughter chromosomes. The spindle fibers are attached with the centromere of the chromosomes. The third stage is the anaphase 2. The daughter chromosomes move towards the opposite poles due to the shortening of the chromosomal microtubules and stretching of interzonal microtubules of the spindle. The fourth stage is the telophase 2. The chromosomes reach the opposite poles. The endoplasmic reticulum forms nuclear envelope around the chromosomes, and the nucleolus reappears. After the karyokinesis, in each haploid meiotic cell, the cytokinesis occurs, and thus 4 haploid cells are resulted. Now let's come to the crossing over. The linked genes that are present on the same chromosome are sometimes separated and form new combinations. The process that produces new combinations or recombinations of genes by interchanging of corresponding segments between non-sister chromatids of homologous chromosomes is known as the crossing over. The phenomenon of crossing over generally occurs during the prophase 1 of meiosis, but sometimes in some animals like Drosophila, the somatic crossing over is also occurred. The mitotic crossing over is very rare and its effect is genetically not significant. The meiotic crossing over takes place during gametogenesis in post-replication tetrad stage. It involves the breakage of each of two homologous chromosomes and exchange of parts. The crossing over includes the following processes. Number 1 is the synapsis. Synapsis takes place during zygoting. The pairing may start from the end of the chromosomes and continues inward in a zipper-like manner, or may begin in the internal region and proceeds towards the end of the chromosome. This is the prolonged and close contact of the homologous regions of the chromosomes. The pairing is facilitated by a highly organized proteinaceous structure called synaptonymal complex. Now let's come to the second process, that is crossing over. At the pectitin, the already duplicated chromosomes of the pairing homologue is longitudinally split as two sister chromatids which remain held together by a centromere. The two non-sister chromatids first break at the same level at the point of pairing. Then, the segment on one side of its break connects with a segment on the opposite side of the break so that the two non-sister chromatids cross each other. Thus, the crossing over includes the breaking of chromatid segments, their transposition and fusion. The last process is the terminalization. At diplotin, this synapsis begins with the dissolution of synaptonymal complex. The chromatids separate progressively from the centromere towards the chiasma, and the chiasma itself moves in a zipper fashion towards the end of the tetrad. This movement of chiasma is known as terminalization. Thus, the homologous chromosomes are separated completely at the end of the dikinesis. Now, let's look at a comparison between mitosis and meiosis. Number 1. Mitosis occurs continuously in the somatic cells, while meiosis occurs in the germ cells. Number 2. In mitosis, its nuclear division is preceded by an interphase stage, while in meiosis, one interphase is followed by two successive nuclear divisions. Number 3. The prophase of mitosis is of short duration and includes no substages, while the prophase of meiosis is of longer duration and it completes in 6 successive substages. Number 4. The process of synapsis, crossing over, and chiasmata formation do not take place between the homologous chromosomes in mitosis, while synapsis, crossing over, and chiasmata formation is done between the homologous chromosomes in meiosis. Number 5. In the mitotic metaphase, the chromatids occur in the form of diheds, while in meiotic metaphase 1, the chromatids of the 2 homologous chromosomes occur as tetrads. Number 6. In the mitotic anaphase, the chromosomes have single chromatid, while in the meiotic anaphase 1, the chromosomes have 2 chromatids and single centromere. Number 7. In mitosis, the telophase always occurs, while in meiosis, the first telophase is sometimes omitted. Number 8. A diploid cell produces 2 diploid cells by a mitotic division, while a diploid cell produces 4 haploid cells by a meiotic division. Now, let's come to the conclusion. For the continuity of a cell and perpetuation of an individual, cell division is required. The division of the cell may be for the purpose of the growth of the individual or for the replacement of cells lost to wear and tear or for reproduction. Thus, the cell division may be mitosis or meiosis. Mitosis results in the production of daughter cells which have chromosome as that of the parent cell, both quantitatively as well as qualitatively. The process of meiosis is much elongated in comparison to mitosis. It consists of 2 nuclear divisions after the interphase that results in the production of daughter cells having half the number of chromosomes present in the parent cell. During meiosis, the exchange of genetic materials between the non-sister chromatids occurred that led to the recombination of genes. It is genetically significant that it increases the chances of variation which is necessary for natural selection and evolution.